Welcome everyone, and thank you all for joining the 8th European Universities of the Seas CU Blue Talk session. This event is organized as a webinar. It's a little bit different than a typical meeting. You won't be able to directly interact with us, but you are free to send us a message in questions or question in questions and answers section, and you are free to chat. And here I invite you all to drop your name in chat and say where are you from, or just say hi. Just please keep in mind that this is Zoom, so you need to click manually in a chat panel that you are sending the message to all panelists and attendees before you send a message. The main connecting medium of the CU Alliance is the sea itself. CU is the glue that binds education, research, business, society, and environment in order to answer the future challenges of the sea. Blue Talks is a series of short expert talks targeted at the broader academic population and representatives of the industry and other stakeholders interested or active in the blue economy sectors. And the idea behind the Blue Talks is that we are looking to entice interest in the blue economy, to raise awareness, and to help you all in understanding the scope of the blue economy. With the first several events, we covered a range of topics for tourism behavior to research and governance in different parts, in different seas, Mediterranean and Baltic, operational oceanography, etc. Today I have a great pleasure to welcome Professor Dr. Alexa Klettner from University of Kiel. Sorry. She's a professor of experimental retinology at the University of Kiel and adjunct professor of experimental ophthalmology in Egypt. She studied biology at the Christian Albrecht University. She did her PhD in pharmacology, working on neuroprotection and habilitated in experimental ophthalmology. And she will give a talk about biomarine compounds as underexplored resources of high potential for development of products for health and well-being. Alexa, thank you for joining us. Welcome, and the floor is yours.
Oh, thank you so much. I just not scare, but share my screen. I hope you can see it all. And
sorry, Alexa. Sorry, Alexa. I have our vice rector finally managing to connect.
Okay, so, I stopped my presentation.
Yeah, just give us a second.
Yeah, sure. I'm happy to see you.
Welcome, Leandra. You could unmute yourself, please. She's muted. Please, Leandra. Yeah, yes. Perfect. We can hear you now. Welcome.
Well,
I just did an introduction so you could cover a little bit about CU and the reason why we are doing this.
Yes, so our European University of the Sea Alliance connects our 6 coastal universities, University of Split, Malta, Cadiz, Brest, Kiel, and University of Gdańsk. And we all share the same vision and mission and want to create a pan-European institution that connects research, education, innovation with contribution to society and to environment. And of course, because of our coastal alliance nature, we are very much focused on sustainability of our environment and on our contribution to the blue economy. And as we all know, important part of the blue economy, actually the emerging sector of the blue economy is blue bioeconomy. And today we have an excellent speaker in this respect, and we are very happy to have the opportunity to discuss the future research trends, innovation opportunities. And I don't know what Nicola all said in the introduction, but I think our speaker today will give us an excellent example of how to go from basic research towards the applications. And I'm sure there are many challenges in the process, and I'm certain that we can all learn a lot from you. There are other research groups in our alliance that are also active in the field of blue bioeconomy, that are also interested in transferring the research results to applications. So I'm sure that they can learn a lot from you. So I know also it's interesting because we want always also to kind of really stimulated the transdisciplinary research and looking from the different disciplines. You're also an excellent example. From what I learned from your CV, you started in biologist, then it was pharmacology, and you're also now in clinical medicine, if I'm right. So it's also a good mix as an example of that. So I'm really happy to have you here, and I hope that all the participants will engage in discussions and also keep in touch afterwards with other questions. So please, welcome.
Thank you so much for this really kind introduction. And I'm actually very much looking forward to interact with you and with you guys here. And, you know, because I'm also a big fan of the transdisciplinary approach. I'm not a clinician, though. You do not want to be treated by me, just to make this sure. But everything else is absolutely correct. So I will start now with the presentation. And I will give you a bit on my experience on transporter cooperation and the development of the biomarine resource Fucoidan. Now, just for an introduction, this is our eye clinic, beautifully located at the Baltic Sea, and this is our research facility where I'm currently located at. And Interreg has been the one that had been financing and still is financing this FucoSan project. So now it works. Great. It's, you know, as Nicola said, we're doing this for so many times now, and we still sometimes have problems with the techniques. Okay, so let's start with the basics. What are Fucoidans? Fucoidans are constituents of brown seaweed, and they are part of the wall, of the seawall of a brown seaweed. And they are very complex polysaccharide, which have a high content of L-fucose, which is sulfated, strongly sulfated. And if you look in the literature, it has a lot of promising biological activities. It's supposed to be antioxidant, anti-inflammatory, antithrombotic, angiogenic, anything. It's also often the opposite. So it's a very heterogeneous compound, and it's very versatile. And you see here one example of the molecular pattern, though I have to say this is just an example because it's very, very much, very different from different species, from different locations, from different extraction methods. And the pictures that you see is actually from our consortium, how we did it from the algae to the Fucoidan. Now, there are many applications. In our project, we looked on ophthalmology, which is, of course, presented by me, regenerative medicine, and cosmetics. And I will take you just for a moment, don't be scared, into ophthalmology, because you have to understand why we are interested in this and what we are actually wanting to do with this. Now, I work on a disease called age-related macular degeneration, which is the main cause for blindness in the elderly in the industrialized world. And to make it very short, because I don't have an hour or many hours to get you into this, this is the eye as Wikipedia sees it, and this little spot here, it's the so-called macula, and this is the point where we have high acuity vision. For example, to see this cute little doggy, which by the way is mine, you need this small part of the eye. And if you go on a cellular level, you have photoreceptors, so-called RPE cells, and beneath this you have vessels. And now what happens when you have AMD, you have, because of chronic inflammation, because of oxidative stress, because of lipid accumulation, the cells start to degenerate. And in the long term, you have degeneration, and you also can have the outgrowth of blood vessels into the retina, and it bleeds maybe, or you have a lot of serum there, and then the cells die and you lose your vision. Now, we can treat this late stage by something called VEGF antagonists, because VEGF is so important for the angiogenic effects here, for the growth of the blood vessels. Now, what we think of, because of all the interesting biochemical properties and bioactivities Fucoidan has, that theoretically Fucoidan could be perfect at this stage, you know, because it could be anti-inflammatory, it could help against oxidative stress, maybe even interact with the lipids, it could inhibit maybe the VEGF, and also, you know, kind of works against the angiogenesis. So our dream is that we find with Fucoidan something to treat an early stage of AMD and prevent the late stages where we actually lose our vision. Now, why I am interested in Fucoidan of all the many compounds that we have? Well, it's because of a mistake that happened in my lab. And I love this actually, because we are nowadays so often forced to give beforehand the results that we have at the end of the project, which is, if you think about it, it's really not the nature of science. Well, what happened here was that my student used Fucoidan for something completely different. We wanted to investigate something completely different and said to me, Alexa, I made a mistake. I'm so sorry. I used the wrong antibody in my Western blot. And I said, why you used, which antibody did you use? And he said, well, I was looking at VEGF instead of whatever we were looking for. And I said, okay, but what was the result? And she said, I don't know, I'll look at it. And it was so interesting that we made a project out of it. She found out that Fucoidan reduced the VEGF in our retinal pigment epithelium cells, reduced angiogenesis. We had a great paper out of it. She got a research award from the German Ophthalmological Society, and all because, you know, she made a mistake in the lab. And I think that's really, really great. So this is why we got started for this. And after we got into this, because of this mistake, I was diving into the literature. And what I found was that Fucoidan had many really interesting properties that made it totally interesting as a therapeutic for blinding diseases like AMD and also diabetic retinopathy, which is the DR, which stands here. I'm not going into that. AMD is complicated enough, but to, you know, to slice this up, it's really a very, very exciting molecule, but it's highly versatile, as I said, and highly heterogeneous. So we have to put a lot of work into it to find a Fucoidan that is perfect for what we're looking for. Now, if you look at product development, if you really want to develop a therapeutic, I mean, this is what bioeconomy is about, you know, you want to develop a product, you have to think that's a pretty long way to actually get a therapeutic out of anything. Because to have clinical application, you have to go to discovery, preclinical development, all the clinical trials, and have to launch it. So this is a long way and very expensive. Luckily enough, in AMD, food supplements are very often used. You can see it here. This is some of the food supplements for the eye that you can find on the market. So this is something you could also maybe achieve or try to achieve with Fucoidan, because this is something that already a market exists and there's an acceptance in the patients. Going on market potential, if you want to earn money, AMD is actually a pretty good call. We have now more than 20 million people affected in Europe alone and 15 million affected in the US alone, which is, you know, the countries that really can spend a lot of money. The global market was in 2015 was at about 5 billion and had a growth rate from like almost 8%. And right now we have almost 200 million people, and there's about 288 predicted for 2050. So we have a lot of patients. And one application of the VEGF antagonist costs like 1,300 euros per shot per application, which is about 9,000 euros a year. But also with dietary supplements, right now patients spend like 150 euros a year. So there's a lot of room for making money here. Now, I'm not into it for the money. I'm into it for the research, but nevertheless, it's always good to know. And we have been, after this mistake happened from my student, and we had this really, really interesting and exciting results, we started, or we established the FucoSan project. And maybe it's also from high interest for you guys, you know, how this kind of international consortiums and collaborations work. Now, our project was called and still is called, we had a prolongation, it's called FucoSan, Health from the Sea. And it's financed by Interreg, which is from the European Union support program for border regions, and this is for the German-Denmark border region. And this is basically where our partners are set. And the project design was actually very cool because we cover basically the whole value chain. We start with the collection of the algae or the cultivation, which was a work package. And the algae we used were many of the Fuco's species, this dictoxiphon, which didn't work, but we used it too. Saccharina latissima, we also had different laminaria species, and they were from different seasons and different locations. And from those, the Fucoidans were extracted and characterized. And the characterization was both chemically and biologically. So we looked at like protein contents, or, you know, what kind of glucose, kind of sugars we had. We looked at the sulfatation, methylation, all these kind of things. And with the biological characterization, we looked like things of like cytotoxicity, proliferation, oxidative stress scavenging, and many others. All the data we have was put in a database. And from the results we had from the basic characterization, we started this pilot projects. And these were, as I said in the beginning, either in ophthalmology, in regenerative medicine, or in cosmetics. And additionally, we also had partners who worked on business models, who looked at the, you know, ecosystem and the potentials that could be done with the Fucoidans. So we had, you know, as I said, the whole value chain in this project. And the only, you know, the only drawback this project has that it only ran the 3 years, because it was really, really good, and we got, you know, great results out of it. Now, these were our partners, and these are basically color coded. Everything that's blue is from Germany, and everything that is, that was green is from Denmark. So we were very close-knit German-Danish corporations with 2 companies joining, which were Coastal Research and Management and Ocean Basis, who were interested. Coastal Research and Management are interested in the algae, and Ocean Basis in cosmetics. But we had also had the Geomai here from Kiel, the Center for Ocean Research. We had the Odense University Hospital, our university hospital. We had the University of Southern Denmark with 2 groups. The Technical University of Denmark, and we had the University of Kiel with 2 groups. So we were pretty well distributed. And if you look at the project design of work packages, this is basically how the partners, aligned to the different parts of the project. So you can see that was really an intermingling with the groups. Now, what did we test? We tested Fucoidans from, as I mentioned before, these different algae species, which came either from the Baltic Sea or from the North Atlantic. And you see pictures here of those that later on I will show you a bit that were the most promising that we had: Saccharina latissima and Laminaria hyperborea. Different extraction methods that were used were hot water extraction, hydrochloric acid extraction, sulfuric acid extraction, microwave assisted extraction, and enzymatic extraction. And I have to ask you not to really ask me anything about the extraction methods, because this was really not my area of expertise. We just used them. As you can see, this I put in just an example of the kind of database. If you're thinking about doing something like this, you really need a very good way of managing your data and make the data available and make the data readable to you. And especially if you, like us, work with hundreds of extracts from different locations and different chemical properties, it's really vital that you have a clear and very good systematic way of cataloging it, so that you know what you're actually dealing with, and can also, at a later time point, always come back to what you actually had worked with. I cannot speak, because I cannot speak for every single project partner, so I thought I would. Well, I was the coordinator, so basically I probably could. But what I mean is that I want to give you the example of the ophthalmological workflow and how we did it and what we did, basically, because this is what I actually know of. So you see here, these are the algae like they were supplied by our partners in CRM. And then our partners that did the extractions jumped in, for example, the University of Kiel or the Technical University of Denmark. And then we had, you know, the extracts which could be tested. And as I said before, the analysis of the Fucoidans would put in this database. We had just to show you some of the basic characteristics that we show. Very important, for example, for our Fucoidans is the fucose content, because that is related to the purity of the Fucoidan, or the molecular weight, because the biochemical properties are very strongly, you know, the bioactivity is very strongly related to the molecular weight. And if we look a bit deeper into the ophthalmology workflow, what we did, for example, we tested our Fucoidans, you know, the many Fucoidans, and we had more than 80 extracts tested in our cell lines, which are interest for us. And we looked at toxicity, we looked at protective effect against different stresses that are all related to oxidative effect, and we would test its effect on VEGF in the cell lines. And those that are interesting and promising in this basic characterization were put into the primary cells that we actually culture from porcine eyes, which are highly differentiated and very close to the real thing in the eye. And here we also looked at VEGF, and what we are also currently doing, not only we looked at function, inflammation, and, you know, its influence on gene expression. And then, you know, at the next level of complexity, we look at organ cultures, which are, you can see them here, which are little organ cultures derived from porcine eyes. And what we are planning to, and we're currently looking for funding, is to now go into the next step and go in vivo, and that is testing our extracts in zebrafish. Now, just to give you an overview, you know, these are the two of the cell lines that we used, and that we had, you know, how many extracts from the different species we had that were protective on the different cell lines. And then we looked at VEGF inhibition. I hope you remember that VEGF is totally important for the development of AMD. And then we have also the different species and looked how many extracts were protective in the different RPE cells that in different cell types. And from this, we basically found our best candidates. And what we found, you know, overall is that our best candidates were laminaria hyperborea. But what we also found is that our best candidates had a high molecular weight, had a high fucose content, and a high rate of sulfation. So we could, in our project, we could really identify what kind of properties we need for our active compounds and which is the best species to use. Our best species were Saccharina latissima and Laminaria hyperborea. And these are the extracts we're currently testing for further further testing, you know, that we could go to the point where we can go in vivo and actually really develop something out of it. There are always challenges, and the biggest challenge actually is that, you know, do you have to get algae of the same quality to get the Fucoidan of the same quality? This is a really big issue if you want to really develop something for the medical application from it. Other challenges is you need a very high amount of the algae. Because of, you know, wet weight algae, you just only have 0.7% of Fucoidan. So it's really vital that you have a sustainable supply of the algae from which you want to make your Fucoidan, because we do not want to destroy we do not want to find a great Fucoidan, but, you know, in order to get is to destroy, you know, the algae banks and the natural resources of the algae. So you need a sustainable harvest. and maybe even think about farming, which, for example, is done for Saccharina latissima. So. And also you have to think about that the environmental conditions of the harvested algae should actually be similar, because this environmental conditions have a strong influence on the Fucoidan structure and therefore on its bioavailability. Okay, so also what we, another output which we had for all of us was creating a database. And if you are interested in, you know, the results, the overall results of the project, we have it published in Zenodo, which is an open access repository. So you can get open access, you don't have to pay to get on it. And it's actually citable, which is great for us scientists. I have to say that we only put the, you know, it only contains data that has already been published in scientific journals, because to protect us and protect our IP, basically, because otherwise other people could, you know, publish with our results. But it's openly available for everybody to see and have a look into it. And just to tell you that it's not only all about, you know, ophthalmology, also the other partners had great results, especially our cosmetic partners from Ocean Bases, which actually developed something they called FucoCrude, which is for their cosmetic prototype, and actually developed a cream during the time of only 3 years time of our project, developed a prototype, which was actually kind of nice because they brought it to one of our meetings, and, you know, it was very smooth on the skin. So I liked it a lot. And to talk about the kind of business models that were developed, it's not like business models developed that we will go into business now and go, you know, pitch to some kind of investor, but what they did was they, you know, looked at where possibly money could be made, at what time this money could be made, because of course it's much easier to, you know, develop food from algae than it is to develop cosmetics, and it's much easier to develop cosmetics out of algae than to develop medicine. But it's also something very important that which they did, and they checked all the patents that were available on Fucoidans, etc. So that was also pretty cool. So if you put together what we did in this 3 really cool but very work-intensive years, I have to tell you, I was the coordinator of this project. So I was basically, it took most of my time during that years. We had 1 violet chains, we had 9 partner meetings. And by the way, partner meetings were in person. This was before Corona. And I have to tell you, if you want to make something like this, if you want to do this kind of big projects that are international, please go meet your people in person. It's really good. We could, you know, a lot of interaction was facilitated there, and a lot of, you know, you also have sometimes some conflicts. You can resolve them there, and it's a really good thing. And it always made people like very excited and very anxious for the project. We had a lot of involved organizations. We had a lot of network partners. We did a lot of networking. I also did a lot of, you know, presenting it. We had 27 scientific publications out of it, which is really cool. And as we had, you know, 19 Fucoidans actually into our databases. So it was pretty cool. And because it was so cool, actually Interact granted us 1-year extension, unfortunately not for research, because they didn't have the money for the research anymore, but for networking. So what we're currently doing, we're in a networking phase, which we have basically the same partners, which a little bit shifting. So what we want to do, we want to use this to go on developing FucoSan-based research projects. I mean, they are going on. I'm currently having another FucoSan research project, you know, which is funded by a different organization, to raise the public awareness of the potentials that are lying here, to identify concepts where we can get more external funding, and to scout, you know, commercial potentials. So what we want to do is, you know, we use the brown algae to take advantage of the Fucoidans, establish value chains, develop concepts, and transfer the results, and strengthen, you know, the bioeconomy in our region. The partners are basically the same. Some shifted from being real full partners to network partners, and we included some other network partners, which is actually pretty cool. And we want to target like you guys, institutions of higher education. We want to target business companies. We want to target, you know, other sectors of like the pharmaceutical industry, cluster organizations. And if you are interested, you can, you know, of course, I hope we have a nice discussion now, but you can always contact me. This is the email, you can contact me, or my other email, which is, you know, my institutional email. And yeah, I hope I haven't confused you too much and there hasn't been too much of input. I'm really looking forward to your questions. And I want to thank very strongly the benefactors for this project. Of course, mainly the FucoSan, Interact with FucoSan, but also Interact Baltic Sea Alliance, which also did a lot to facilitate this. I want to also thank the Deutsche Forschungsgemeinschaft because it was in one of their projects where we first found Fucoidan. I want to strongly thank, oh, I'm sorry, now it's gone. But anyway, I want to strongly thank the Helmut Ecker Stiftung. I want to thank my my partners and my FucoSan partners, and I want to thank my current PhD student Philip for all his work. And now I'm done, and I thank you all for your attention.
Perfect, Alexa, thanks. So it's interesting to see this kind of holistic overview of the whole process. You know, typically people focus on just one thing, then they're doing it the best, you know. So currently we don't have many questions. I would invite everyone, if they have questions, they still have time, you can write them down. The easiest way is to write them down in chat. If you want to be included, we could also add you to the discussion as one of the panelists. But let's start with questions. So let's go with Daria. Is there experimental aquaculture of the Fuco species? Mm-hmm. Do you think that integrated aquaculture of the Fucus and fish will have an effect on the Fucus quality?
Very good question. So that was part of our project, actually. So our partner, Raphael from CRM, he was actually, his task in our project was to try to culture Fucus vesiculosus, which was not as easy as he hoped it to be, or as we hoped it to be. And he's still working on that. And I think I have to, you know, you have to go to talk to Raphael to really see how this works out. But it's the problem is that you have to concentrate on the asexual part of the life cycle, because as soon as the sexual part of the life cycle starts, you know, you have, I think, a decrease in biomass and a decrease in the Fucoidan. So this is a rather complex thing. And also there is always the problems of the epiphytes on the Fucus. So if you want, it's not easy. It's much easier to go, you know, Fucus, if I remember correctly, Fucus is not aquacultured yet. And I think this is good reason for it, because it's pretty difficult to do. As your questions for the fish, if it's, you know, if you put it in a combined thing, we have not tested that. But I think that Raphael is actually currently putting up, or at least the company Kieler Meeresfarm is actually doing something like this right now to combine the, you know, the algae cultivation with the fish cultivation. But I think they're using Saccharina. And as for your questions, we actually would have to test it, whether it has any effect on the Fucoidan, because we know that Fucoidan is, you know, salinity is a factor. Probably even, you know, the how you say this, you know, the strength of the ocean is a factor. You know, if it's quiet, or if you have a lot of movement in the sea and all these kind of things. So we would have to test this, if it has any influence.
So to continue on this, you basically source the algae from two different sites, from two different seas, from Baltic and from North Atlantic. So what kind of differences have you seen there?
This is the thing is that the original idea was to harvest the same species from different locations. We have some, from example, those are harvested Fucos. We have Fucus vesiculosus harvested from Denmark and from Kiel, but it's very difficult because of environmental regulations, because you are allowed to do this in Denmark, but not so much in Germany. And what we found is that the two species that we got from what we found is that the two species that we found best were both from the Atlantic. But I don't know if it's really because they're from the Atlantic or because of the species, because we did not really compare, for example, Saccharina latissima from the Atlantic and the Baltic, which would have been great, but we just didn't. We didn't have the opportunity to compare them. So for us, we see that those from the Atlantic are better, but it could just be related to species and has not so much to do with the origin. Though I have to say, after attending many algae meetings, which I'm not naturally an expert in, because I majored in zoology and gave up botany as soon as I could back when I was a student. What I learned is that the yield in the Atlantic is also much higher. You know, the Saccharina grows much better in the Atlantic than it does in the Baltic. So probably the Atlantic is a pretty good source for our algae.
Yeah, we have one more question connected to this from Martina. Martina is our PhD student, and she's interested basically about the possibility of working together, maybe on identification of Fucoidans from Adriatic Sea. Martina, would you like to? I could connect you also, or if just say yes in chat, and I will connect you. You could maybe ask a few questions live. But Alexa, yes. Please.
Oh, I would just say, yeah, I would be delighted to test the Adriatic Sea. I think it would be a great idea. I don't know, I'm not familiar with the species that actually grow in the Adria, but I would be delighted to talk about this. And if there's any chance to do that, I would love to. I just have to add, I cannot extract Fucoidans. You know, if you sent me algae, we would need to find a way to make Fucoidans out of the algae in between. But I would love to, yeah.
Perfect. So no questions, but I have one, and I probably think that Leandra has some. By the way, she's smiling. Maybe you, Leandra, and then if you have.
Okay, well, I'm neither algae nor ophthalmology is my expertise, but so I'm wondering, just in general, how long would it take for you to arrive to the, some commercial applications in the field of ophthalmology? You said the process is very long, but you have an estimate.
Yeah, well, this is a big if I were to really want to develop a medical product out of Fucoidan, and would ignore the obstacles like where do I get the funding for my clinical studies, and where do I get a Fucoidan that is clean enough and chemically, in a way that we could reproduce it in a way that we can actually get an FDA or an EMA approval. If I not think about this, it would be generally, they say, from the idea to the therapeutic, it's 10 years. 10 to 12 years, and it costs millions. So, currently I'm doing this research, and if I find something that's really cool enough, I could go and, ask, you would need at a point, just to say, you would need a pharmaceutical company that is interested. But if we're looking at dietary supplements on the way, this can be done in a few years' time. The question is whether you want to have a health claim or not. If you want to have a health claim, even for dietary supplements, you need some kind of clinical data. It's still, way. If you do not want a health claim, you can do it very fast. But, I wouldn't want to do this. If I really want to develop, bring something on the market, which I stand behind with my name, I want it to be, you know, good, really good, so that I probably would want a health claim. But this could be done in some years' time. This is not so far ahead.
Yes.
And I also think, you know, if it's really,
it could be helpful, yeah.
Yes, yes. And if you know, the thing about, I mean, for me, you know, just to make it, AMD is a multifactorial disease. We have many pillars on which the pathology stands. And usually what we have nowadays in therapeutics is we have, you know, one therapeutic for one pillar of the disease, like the anti-VEGF. And the big potential of Fucoidan is if you find the right one, you can target many of the pillars at the same time. You know, and currently we're having a Fucoidan that is anti-VEGF, and we find it to be anti-inflammatory and maybe even antioxidative. You know, and if we find the perfect Fucoidan, I think we will get people interested in developing something out of it, because this is something that current therapeutics do not have. They are just so single-minded and just one pathological pathway. And this is why I'm so excited about this substance.
Oh, it's very interesting. Yeah.
Thank you.
Yes, thank you.
No more questions in chat, but I was also, because you had a whole business model like in stages, and you had nutraceuticals, and is that I understand your answer, but is that a way to fuel the future research and fuel all the following stages? So even, yeah, sorry.
No, please.
No, I just was wondering how this kind of, it's a complex project, you know, it's a 10-year, you have a 10-year goal. It's not an easy target, you know, to do something for 10 years, and there's a lot of point of failure, you know, there's a lot of opportunities just to miss the target. So I was wondering, how are you looking at this, all opportunities that you could have? Do you have any kind of support, you know, from your university, or do you have any kind, or you are also depending on the projects and your, you know, from day to day or from project to project basis?
Yeah. This is two questions, basically. The first
it's a lot
is
of
with
questions.
Yes. Well, I will narrow it down to two, and then if I miss anything, you can ask the others again. So the picture that you saw was for all partners, you know, with the different opportunities. And for me, you know, this is not all for me, but for the different partners. I think this is very important for the FucoSan project, and this is why I was presented it like this, because it's a team effort, and it's just the different points where Fucoidan could be made into a product, you know, with at a different point on the value chain. But I wouldn't be the one who makes the money out of it, but the other partners maybe. And so you could maybe cooperate with them. As to your questions with the development, I am on a project to project base, like most of us are. You know, we are on a project to project base. I do not have a great scheme grant or strong support from the university, which sounds unfair. I mean, my university, of course, supports me, but only the way it can. So it says, get your own grant money. And this is, you know, the way it is today. But we have the next grant money, and I'm working on the next grant money. And, you know, and what you said about failure, you know, I think this is one thing in science that we have to, you know, that you have to get used to it, or you have to leave science, because you need to cope with failure. And as some great person said, you know, fail next time, fail better, you know, fail often, fail better. And it's the thing. And if we find out the Fucoidans will not become a therapeutic for AMD, we still have found out a lot about their properties, and somebody else will find, you know, and whatever we find out with them will help somebody else to make something out of this, you know, and it's, I know, I'm actually pretty relaxed about this. I mean, you know, that's a good thing. If you're a tenured professor, I don't have to worry about, you know, what I eat next day, but just about, you know, and I always think that it will go on, because it's great data. We have great publications. We just got, you know, a review out with colleagues on marine products, and it had like an impact factor of like more than 7. And if you come from my field of research, this is really a lot. And you know, there's a lot of interest in this. I'm invited to go to Russia in August. I hope I can, you know, because of Corona, to present all this. And So I'm actually very confident that if we fail, it's life, it's science. Just have the next great idea.
Sure, and I think this is great what you said. You put all the data in open access afterwards, and so who knows, maybe someone else will see something else in this data that you don't see, and the things can go on. So that's great.
And also it's a question how to combine the open data and open science with managing IP, intellectual property, and especially in this type of long-term projects. Sometimes you would need to protect something quite early, even if you don't perceive what will develop out of it. So it's always a question, and it's a really money-intensive thing that sometimes it's not recognized, even through projects or whatever. So it's a huge challenge, I imagine.
Yeah, I was a bit naive when we started this kind of things because, you know, I just put my data out and later I was told, why didn't you put an IP on this? And I said, well, no.
What's that?
Yeah, I'm a researcher. Why should I? And they say, you know, this is has really potential. I said, okay. So, but on the other hand, you know, it's like it is, and there will be always, if there is something, you know, I think there's a product in there, and even if, you know, other people could also make a product out of it, I mean, that is, if they want to invest the time and money to do this, we can cooperate.
Perfect. Thank you for everything. Thank you for your optimism and bright approach to the topic today. Even if it was complicated for me, it was really interesting to see how the way you presented it. So thank you very much, and I hope that we will have opportunity to cooperate in future.
Thank you. Thank you so much for inviting me, and I hope it was not too complicated, and I'm really looking forward to cooperate both scientifically and if you know, with this teaching, which is also, you know, if there's any opportunity to me to go on to develop this further, you know where to contact me. I would be delighted. Thank you.
Yeah. Great. Thank you very much.
Thank you. Bye-bye.
Very inspiring. Bye-bye.
Bye.
Great.